and I will discuss, the lectures for the clinical chemistry. So the first thing that I will start it's about this safety and about the safety regulation that is present in the Germany. So we have these are two regulation that is important to know um, about how it's safety, how safety handling with the organisms or what stuff with the biological that is present in the Germany. So we have the first one is called Biostoff V and Gene TSV, and we wanted to know what they are related to. So the first one is Biostoff, and Biostoff it's a German word, and that is mean the organisms, the microbes. I mean here the bacteria, the viruses, the other organism. And V that's mean Verordnung, and the Verordnung it's mean recoagulation. Okay, and we have another one is Gen TSV, and that is mean gene, that's mean from genetic, T technique as Sicherheit, and Verordnung that's mean the Verordnung for the genetically modified organism. So a general idea about these things, the Biostoff V that is deals with the biological organisms, where the Gen TSV that is dealing with the genetically modified organisms. Now we're making just a comparison between the Biostoff V and the Gen TSV. So the Biostoff V, again, V that's been recalculation in English, Verordnung in German, and that's mean a targeted assessment. Target assessment, and this type of the assessment looks up non-risk when working with the biological substances. That's why we said the target assessment, because it's type of the assessment that like looks and searching what are the risks that we dealing with when we dealing with the biological organisms, or we can say it's the safety measurements and safety, safety things that we have to do it when we are just dealing with the bacteria, with the viruses, with the other organisms. So this type of the assessment looks about the risk when we are just handling with the microbes like bacteria, viruses, and naturally. Okay, so there are three criteria for that. The first one is the species involved is identified. The focus is directly on the specific biological compounds. There is a recognized or potential risk of exposure to the workers during the normal operations. The first one, the species so involved is identified. If I said there is an organism or there is a bacteria that will cause risk for the workers, for example, so the first one I have to identify which bacteria I have to, that causes the risk to the workers, and the other thing, the focus is directly on a specific biological compound and specific things, like more specific on these things. And then we have to identify what risk, what recognized risk that the workers exposed to when they just dealing with these organisms. So the first one, the species identified or should identify, the focus is specified on specific biological compounds, and the other thing is identify what is the potential risk that is can affect these workers during the normal operation. So we have another criteria, it will be the same: identification, evaluation of the risk, determine protective measures, and measures or insurance the safety during the workplace. So I think we can say the most important thing that you have to know it is the Biostoffverordnung. It's target assessment. This is another name. So it's like identify what is a potential risk when we're dealing with a biological organism like bacteria, viruses, parasite, etc. Okay, so the first one, the species involved should be identified, should focus on specific biological compounds, and also evaluate and identify the potential risk of the workers during dealing or during their normal operations. So these all about the Biostoff, and you have to know also that there are four risk groups. Like one is the low risky group and 4 is the high risky group, and we will discuss it later. But the risky groups is under the Biostoffverordnung. So that is talking about the PPE, so that how to dispose the safe waste products, the vaccination, all they are under the Biostoffverordnung, healthcare, architectures, waste management, labs, okay, and handling with the infection samples and the hospitals and all this stuff. Then we go to other regulation is called GEN TSV. And the GEN TSV, it means here that it's genetically modified organism. Another name for it is risk assessment. Okay, so evaluate the potential risk associated with the genetic engineering and genetically modified organism. Like we change this bacteria or we add something to this bacteria or to this organism. So we are dealing with a genetically modified organism. I think that is the most important. When I said Biostoff V, that's mean we are dealing with the biological organism, while when we say GEN TSV, that's mean we are dealing with the genetically modified organisms. That's it, okay? And we have to know Biostoffverordnung, that is target assessment, while the GEN TSV, that is taking about what we can say it's a risk assessment. So it's evaluate the potential risk that is associated with the genetic engineering and genetically modified organisms. So it has a 5 criteria. The first one, identify the source of the genetic material, like the donor. We are taking this genetic material from something, so we have to identify what is the source of the genetic material, and then understand how the gene is transferred. So we have to know what is the vector, how this gene is transferred, and then evaluate this gene that is being transferred, and then assess the recipient, the acceptor organism, and then classify of all these genetic modified organisms. So I think it's be logic how what are the criteria that is associated with the genetic TSV. Identify the source of the genetic material, understand how the gene is transferred, and then evaluate the gene that is being transferred and assess the recipient, and then classify what is the type of the genetic modified organism. And again, here we talked about the Gentechnik-Sicherheitsverordnung. That means it's a genetically engineering. So they are subdivided into 4 safety levels. So we have 4 is the high risk, and S1 is the no risk. So that is the subdivided of the genetically modified organism. So again, I will repeat 2 things. We have the Biostoff V, its target assessment, and is associated with the or determined what are the risk that is associated with while we handling with the biological organism like bacteria, viruses, and etc. And the criteria, we have the species that involved should be identified, should focus on a biological compounds and specific biological compounds, and the other things should be determined, evaluate, and identify the potential risks that exposed to the exposed workers during their normal operations. And it's subdivided into 4 risky groups, and we know that the 1 is a low risk and 4 is a high risk, and we will discuss it later. And then we have the GEN TSV, and that is risk assessment, not target assessment, risk assessment, and that is evaluated the potential risks that is can be results from the genetically modified organisms. So the criteria: 1, identify the source of the genetic material, the donor, understand how the genetic materials are transferred, the vector, and then evaluate the gene that being transferred, also classify assess also the recipient, and classify the genetically modified organism. Okay, so these all about the Biostoff V and GEN TSV. Hope you understand this part. Now we go to the risk groups, and these risk groups, as you can see here, they are so important. These are risky groups. We can say they are subdivided, or they are under the title of the Biostoff, because we are talking about the biological organism. We are not talking about the genetically modified organisms. Okay. So the first one is the risky group 1, the biological agent, not cause. to the human or vibrates any disease. So the biological agent, whatever is it, bacteria or viruses, whatever, not causes any disease to the human or the vibrates or animals or anything. It is spread to the community, no spread to the community. Wild type, the examples for that, like wild type bacillus subtitles. So the bacillus subtitles, it's one of this type that doesn't cause any disease to the human, the vibrates to the animals, doesn't spread to the communities. Okay, that's good. So there is no risk to the community and low risk to the human. And we can say it's a biosafety level 1. So when I say biosafety level 1, I mean it's safe, okay? So this is the risky group 1. Now we go to the risky group 2. It causes disease to human. It may be causes to the worker, but it's moderate risk to the individual. Unlikely can be spread to the community, okay? But low risk community risk, okay? And the examples of that: wild type E. coli, measles virus, Epstein-Barr virus, and we will discuss what is Epstein-Barr virus. But these organisms, I mean wild type E. coli, measles virus, Epstein-Barr viruses, causes disease to the human. They spread to the community, but it's low community risk but moderate risk to the human. That's all you know, and this is the biosafety 2. The risky group 2, that causes disease to the human, causes disease also to the community, spreading to the community, but it's high individual risk. It is still low community risk. And the examples for that: Mycobacterium tuberculosis. And then the risk group 4, it's causes severe disease to the human, serious diseases to the human, high risk of spreading to the community because there is no effective prophylaxis, no treatment available, like Marburg virus and Ebola virus. It's high community risk and high individual risk. So all these risk groups that we are talking about it. So let's focus on the risk group 1. Risk group 1 doesn't cause any disease to the human or causes any spread to the community. So no disease to the human, no spread to the community. The best example of it is wild type Bacillus utilitas. Okay, so there is no risk. Then we make a comparison between the risk group 2 and risk group 3. Both of them causes disease to the human. Both of them separate disease to the community. But what are the difference? Both of them causes low risk community, but the difference that risk group 2 causes moderate risk to the human, to the individual, and risk group 3 causes high risk of the individual. Now the example: risk group 2, like wild type E. coli, and also the measles virus, and also the other thing is Epstein-Barr virus. So we have the wild type E. coli, the measles virus, and also we have the Epstein-Barr virus. They are everything, it's okay, but moderate risk to the human. And then we call the risk group 2, like Mycobacterium tuberculosis. It causes high risk to the individual, but still low risk to the community. And then 4, it causes everything severe disease to the human, severe disease to the virus, no prophylaxis, no treatment. And the most important examples for that is Marburg virus and Ebola virus. It's important to know that the wild type E. coli, measles virus, and also Epstein-Barr virus, they are under the group 2. Wild type Bacillus utilitas group 1. Mycobacterial tuberculosis group 3, and group 4, it's Ebola and also the Marburg virus. Okay, so that's important to talk about these things. And we have the risk group 1, we can say it's a biosafety 1. Risk group 2 is a biosafety 2. Now we talk about something so important to know. It is called E. coli DH5A, or we can say K12 strain. So what is it? Why we called it? It's actually E. coli DH5A. It's a genetically modified organism, okay? Genetically modified bacteria. It's a new DNA that we transferred it to the bacteria. Genetically modified organism, but it's under the biosafety level S1. It has low risk. So we added a DNA or the vector, but when we just make a new operations on the DH5A, we add more risk. Okay, but I wanted to talk more about this. Okay, so it's called E. coli DH5A. Why it's called DH? Now we understand it's a genetically modified organism, but the question is always why it's called DH. It calls DH because it's referred to the scientist is called Douglas Hanahan. Okay, and the Douglas Hanahan who discovered this E. coli, not discover it, it just like make a modifications in this coli, and that is referred to it. So it's called Douglas Hanahan. Why it's 5A, I don't know, but it's referred to the Douglas Hanahan. Okay, it's a genetically modified bacteria. Why we use it? Use it for cloning. Okay, that is used for cloning. And the questions, what is mean exactly by the cloning? It's make many copies of the plasmid. Why we make many copies of the plasmid? Because we needed it for sequencing. So again, E. coli DH5A, it refers to the Douglas Hanahan. It's genetically modified organism. We use it for a cloning application. That's mean making many copies of the plasmid, which is important for the sequencing. Good. Then the other information that we have to know is how that is can be happen. Like this bacteria, which I mean E. coli, then we put it in the calcium chloride, or we can make electricity. Then we make these pores wider. Okay, then when it's these pores wider, okay, we make, because we wanted to throw these pores, this genetic material to go inside it, so we incubated it into the ice. Okay, so incubated it to the ice to make this DNA material so very close to these pores, and then we make a heat shock. Why we make a heat shock? Then this, by the heat shock, these genetic materials, it will be inside this bacteria, and then we make recovery at the 37. So this genetic material, it will be inside this bacteria. That's exactly how we insert this DNA inside the bacteria. And because of this mechanism, it's an important in something is called the blue-white screening. What this is a blue-white screening? The plasmid containing something is called IAZ. IAZ makes it looks blue. Okay, when we just insert the DNA, if inserted very well, then it will break IAZ, and when it's a break IAZ, then it will appear white. If it's not inserted well, then it will not break IAZ, and that it will still blue. So it's an important in a blue-white screening. If detect, it's an important in detecting the DNA of insert. It detected if it's inserted well. This is the first, and we can just observe how our DNA growing. That's fine. Okay, then the other thing I have to talk about also the DH5A, that is stored in cholesterol. And the question is why we stored it in the cholesterol. This is a big question, because we are stored into two conditions: low temperature and the cholesterol. Low temperature, it means it will be, cold. But the cholesterol, and the important thing about the cholesterol, that is depressing the freezing point of the bacterial cell. So it will be cold and enhance the supercooling. At the same time, it reduces also the harmful effect of the ice crystals. Okay, so 2 points. The first one is depressing the freezing point of the bacterial cell, that is enhances the supercooling. And the other thing which is so important, that is prevent the harmful effect of the ice crystals. So I think, yeah, I just said a lot of informations about the E. coli DH5A, but I have to know all this information to be able to understand what is it. Okay, so we understand the E. coli DH5A, genetically modified organism. This is the first thing, and the most important thing, that it's used also for cloning, making many copies of the plasmid, that is used for the sequencing. And we know that it's referred to the DH Douglas Hanahan 5A we know how to insert the plasmid or the DNA of interest inside it by just the first thing that we make calcium chloride and electricity to make the pores wider. Then when you incubate it into the ice to make the DNA very close to these pores, then we make a heat shock, then the DNA will enter inside the bacteria, and then recovery, and then we do our thing. It's an important also in a blue white secreting, and we understand that the plasmid contains IAZ, which is make it blue. If inserted well, then it will turn white. If it not inserted well, then it will still blue. Then we just recognize it to be inserted well, and just we observe the growing of the DNA. We also stored it in the glycerol for 2 points. The first one is the the freezing point of the bacterial cells, so we enhance the supercooling, and the other thing is protecting these bacterial cells from the harmful effects of these icy crystals. So we clean the lab surfaces with the Bacillol AF. Now we talk about the chemical methods of the microbial control. Antiseptic or disinfectants like phenol, alcohol, halogens, heavy metals, detergents. So we use these antiseptics or disinfectants like phenols, like alcohols, like halogens, like heavy metals and detergents. We can use also the chemical strains like ethylene oxidase, propyl acetone, glutaryl dialdehyde, and formaldehyde. Okay, we use also these are, we can say, is the chemical methods. And the questions: antiseptic, disinfectant, we know that phenol, alcohol, halogens, detergents, heavy metals also, it's important for the cleaning. And we know also ethylene oxidase, propylactone, glutaryl aldehyde, and formaldehyde. We just enough save in our mind or memorize ethylene oxidase and formaldehyde. We can protect microbes also by radiation. So the radiation is subdivided into ionizing and non-ionizing. So ionizing like a gamma rays, X-rays, and also high energy electron beam. Gamma rays is ionizing, X-rays is ionizing, high energy beam is ionizing. And you know, that is why the pregnancy or some individuals they don't have to expose to the X-ray, okay, because they are ionizing, because they are dangerous. And the same, we use it also to protect our surfaces. We use the gamma rays, we use the X-rays, we use also the high energy beams. But these ionizing, non-ionizing, ultraviolet rays and microwaves. So about the ultraviolet light, I mean here the non-ionizing. Ultraviolet lights decontaminate the surfaces, and the materials do not absorb the light. So it's like clean all the surfaces that they cannot absorb the light. Decontaminate and disinfected the surface do not absorb the light, like water, like air, and these things. Microwaves kill the microbes indirectly by hot. So the microwaves is kill all the microbes but indirectly by hot. Now we go to the Epstein-Barr virus. Now we discuss all the information that is present here, and I want also to add some information. The first one is enveloped virus, and I said in a blue, so we have inactivated it by detergents. That's why we use alcohol, phenol, detergents, halogens, etc., heavy metals, because it's enveloped virus. So it's protected, but we have all these protections to inactivate it. And that we can inactivate it by the detergents, by bacillol, by Clostridium. So the first thing that the Epstein-Barr virus is enveloped virus, so we inactivated it by the detergent bacillol and Clostridium. The other thing that is DNA virus, it contains double standards of the DNA. Okay, the other thing that is, I want also to add something that is Epstein-Barr virus. The other name of it is a human herpes virus 4. Okay, so the other name for it is a human virus herpes 4 herpes virus 4, you know. And the other thing that is infect beta cells and epithelial cells. Okay, so it's infect epithelial cells because it goes to the nasopharyngeal mucosa, and after it's affecting the nasopharyngeal mucosa, maybe causes the infectious mononucleosis or nasopharyngeal carcinoma. That's we hear it. All these diseases that are associated with the Epstein-Barr virus. But let's focus infect the beta cells. We know that the beta cells, B cells, sorry, or B lymphocytes, they are just a part of the blood. So Epstein-Barr virus bind to the CD21 receptors. These receptors are present on the surface of these B cells. Again, B cells, there are receptors that is present on the surface of the B cells is called CD21, and the Epstein-Barr virus binds to these receptors and then making it immortalized, or something that is happening to these B cells. They are not working well, okay, and that is increasing the level of the monocytes, and after increasing the level of the monocytes, that is causes infectious mononucleosis. Okay, and also affect the epithelial cells, affect more than 90% in the first 2 decades of the life, actually affects in a 20 in a 10 years. And the transmission that is can maybe happen by the saliva or by the sexual contact. That's why we see kissing disease or glandular disease or this stuff. The transmission period is 4 to 7 weeks. Some say 4 to 7 weeks, some say from 3 to 6 weeks. So the numbers are different. Is there a vaccination for the Epstein-Barr virus? No, there is no vaccination of it. Sometimes it's asymptomatic and sometimes it's not. It's like a flu, malaise, headache, low-grade fever, sore throat, tonsillitis, lymphadenopathy, especially the posterior cervical lymph nodes that is affected it, and maybe that is lead to the more complication and causes splenomegaly, hepatomegaly, decreased appetite, arthralgia, photopia, skin rash, diarrhea, and these things. And maybe it's maybe gone by no treatment after 3 weeks or 2 weeks. Okay, and sometimes they give you a cyclovir as a treatment because it just inhibit the replication of the Epstein-Barr virus. And maybe developed infectious mononucleosis, because if they just ask you what is the cause of infectious mononucleosis, Epstein-Barr virus. When we just mention the Epstein-Barr virus, we mentioned it in the risk group 2. This causes disease to the human and causes disease spread to the community, but low risk community and moderate risk to the human. It's important to know that it's associated with the two tumors: Burkitt's lymphoma, nasopharyngeal carcinoma, and also Hodgkin lymphoma. It's also associated with this virus. Again, I will talk about some information, like it calls human virus herpes 4. It's the same of Epstein-Barr virus. It calls kissing disease because the transmission it will be through the saliva and the sexual contact. Sometimes it's called glandular disease because it's affect the lymphocytes and the glands, so especially the posterior cervical lymph nodes. That's why we see glandular disease. Overactive immune system or lack of immune system. It associated with the beta cells, as we said, that is bind to the CD21 receptors that is present on the surface of B cells, and that is causes immortalize. It may be asymptomatic. The incubation period it's from 4 to 7 weeks. What else? It's especially affected the oropharyngeal epithelial cells. We are talking about the septum. We are talking that is a DNA because it composed of the double standard DNA. It is present in the nasopharynx, oropharynx, and the tonsils. It also the bloodstream. It's composed of the lymphocytes, cytotoxic. Okay, and there is no vaccine for the Epstein-Barr virus, and sometimes we give a cyclovir that inhibits the replication of the Epstein-Barr virus. And now all this stuff that is associated with the lecture 1, hope you understand anything, and the upcoming lecture, it will be the lecture 2 of clinical chemistry. Thank you so much.